Okay, great. And thanks so much, Sean, for agreeing to speak at such short notice. It's really a pleasure to have you here. Just for those of you who aren't familiar with you yet, so Sean leads the Cognition, Anatomy, and Neural Networks research group at the University of Bristol. And he originally did his PhD, as we were just saying a moment ago, in King's College London on neuroimaging. I think it was a very dauntingly productive PhD. I won't say how many papers you wrote, Sean. Um, and that was followed by a couple of postdocs in New York, at NYU and also at Mount Sinai. And yeah, we look forward to hearing about the work you're doing in Bristol now.
Yeah, thanks so much for the invitation. It seems like a fantastic group of people, and I look forward to discussing with you and learning from you. So yeah, I'm just here, I suppose, representing my research group. They're, these wonderful people here. A lot of us are in Dublin now for the RLDM conference, so it's nice to step away from that for 1 hour, one hour, and speak with you, and then back into conference mode. So yeah, like Petra said, when I started in neuroscience, I was in neuroimaging, and everybody was very excited about the connectome. And if we just understand the connectome, that will tell us who we are, right? It will tell us about mental illness, about cognition, etc. And I fell in love with this idea. I went and I studied everything I could learn about white matter anatomy. Um, and I started later to learn about nonlinear dynamical systems and how, well, the connectome really determines the dynamical landscape of the brain, or the patterns of brain activity that it's easy for the brain to produce and to maintain. Okay. And the analogy that's usually given here between patterns of neural activity and nonlinear dynamics is that of a ball rolling down a hill, where each position of the ball on this landscape corresponds to one particular pattern of neurons that are active in the brain. And just like there are certain positions in this landscape, the valleys that it's easier for the ball to get into and be maintained, there are certain patterns of brain activity that it's easier to get into and be maintained, right? And in both these situations, we can call these attractor states. Okay. But then I was very influenced by someone who's now my collaborator, Amy Arnston, this wonderful neuroscientist at Yale. And Amy described this concept of dynamic network connectivity, which is basically a series of observations from her studies and others that neuromodulators, things like dopamine, noradrenaline, acetylcholine, can cause these transient changes at the spines, at the synapses between neurons, so that for a brief period of time, the strength of the connectivity can change, right? And Amy really focused on and described this process in dorsolateral prefrontal cortex in the brain, a really important area for higher cognition. But I suspected that this process might be taking place also in other parts of the brain. Um, and uh, and by having this process occurring throughout the brain, we may start to think of the brain's dynamical landscape not as some sort of a fixed hills and valleys, but more of a moving landscape, if you like, something more akin to the hills and valleys of a duvet in a bed with a person in it, right? And you can imagine that depending on the positions of somebody's arms and their legs, it might be easier for a ball to get into one particular uh, location on the duvet, right? And to bring that back to the brain, that is to say that depending on the pattern of certain subcortical inputs to the cortex, particularly from the neuromodulators, it may be easier for the brain to get into certain patterns of activity according to the type of neuromodulatory inputs that are present at a particular time. Okay. So I was very inspired by Amy's idea, but like I said, it was very focused on dorsolateral prefrontal cortex, and I wanted to know where this dynamical network connectivity might be happening across the brain. So I teamed up with this wonderful neuroanatomist in Germany called Nicola Palomero Galler. And Nicola and her team had just systematically measured across more than 100 cortical regions in the macaque monkey the the density of more than 14 different types of neurotransmitter receptors. Okay? So this will allow us to understand where are the receptors in the brain that enable the changes to the brain's dynamical landscape. Okay. And we had a series of studies, but here I'm principally going to talk about one study where we put together information from as many regions as we could, as many regions as we had data for. Okay. And by first step when we analyze data um, is to visualize it. Okay, and second step for dummies like me is to do a principal components analysis. Okay, and so we did a principal components analysis on this data, 109 regions, 14 types of receptors. Okay, and what we see here is we've plotted all of the data down into this two-dimensional space, which we can call the receptor space. Okay, and we can call it the receptor space because the position of each brain region is determined by the pattern of receptors that neurons in that area express. And two areas are closer to each other in this space if they have a more similar pattern of receptor expression. Okay. And we found the cortex is really organized along two major axes that explained a huge proportion of the variance, well over 80%. Okay. And if we first dig into this first component, this major component on the horizontal axis, we can start by looking at the coloring. So we've colored these areas according to the resting state networks, or some people call them the cognitive networks, that people often refer to in cognitive neuroscience and human fMRI. And we have the sensory networks, here, and purple and blue, and the more higher cognitive networks in these other colors. And what we see is that a lot of these sensory networks, and in fact, almost all of the areas in these sensory networks are clustered towards the left-hand side of this axis, and the more cognitive network areas are towards the right-hand side. So this tells us that this first component seems to be pulling out a kind of a sensory cognitive hierarchy according to receptor expression. So we wanted to dig into that and understand that a bit more. And luckily, we were actually working in the monkey here. We do some work in mouse and marmoset and human, but today I'm principally going to be talking about monkey work. And in the monkey, you have actually decades of neuroanatomy research from different labs up until the present day, which allows us to compare different aspects of brain organization and try to understand this sensory to cognitive receptor gradient a bit better. So, uh, working together with my neuroanatomist colleagues and using some tricks from neuroimaging, we put together lots of different types of neuroanatomy data, and this is just some of that. And we brought this into a common cortical space to try and see if there are some shared organizational principles to this large-scale cortical anatomy. Okay. And the first thing to notice is that this principal receptor gradient, this principal component, really just pulled out the total receptor density. Okay, so that's per neuron. So neurons that brain areas that were further toward the right of this axis had neurons with much more receptors. Okay, but notice also the scale here. I think this is something that is not well known or appreciated in neuroscience. Is that yeah, we estimated that neurons uh, towards this right of this axis, so prefrontal cortex, associated parts of parietal cortex, etc., had about 4 times as many receptors as neurons in primary sensory areas. Okay. So that's kind of interesting and surprising. And so how can they do that? Like what is enabling neurons that we normally think about, pyramidal cells, they're pretty much the same in one part of the cortex and the other. So what can enable this kind of fourfold increase in the number of receptors? And one of the things we looked at was the size of the dendritic tree, a series of studies from Guy Elston and colleagues in Australia and Japan. And we found that the size of the dendritic tree also increased systematically along this axis, and increased about roughly threefold along this axis. Okay, so the way I understand that is you're really given more neural real estate by having bigger dendrites that can allow you to fit in more receptors along those big dendritic trees up towards the prefrontal and parietal cortex. Okay. We then looked at myelin, and one of the reasons is that myelin is, we know it has this systematic gradient along the cortex. It also uh, is a lot of interest in the neuroimaging community as a potential non-invasive marker of cytoarchitecture. Okay, and we found a strong negative correlation between an indirect measure of myelin, the ratio of the T1 weighted to the T2 weighted image, with our principal receptor gradient. Okay, but that is an indirect measure and it's been criticized, particularly outside the cortex. It might not measure myelin so directly. So we went down into the slices and looked at direct myelin stains in three different brain regions. And this is just one example receptor, the NMDA receptor. And here the density is plotted from left to right in this white line, with greater density being more towards the right. And almost all of the receptors showed this kind of a pattern where it was basically anti-correlated with myelin across the layers. In all of the regions that we looked at, and for almost all of the receptors. And so we see this kind of anti-correlation between receptor density and myelin density. And I think that makes sense, right? Because where you have a myelinated axon, you can't have a synapse, right? There. So there's no real place to put a receptor, right? There's not going to be a synapse there. And there's also other preclinical research suggesting that myelin actually has an inhibitory effect on plasticity. Okay. Um, and then the last neuroanatomy thing that we looked at for this gradient was an independent measure of the cortical hierarchy, or say based on independent data. And this was based on retrograde track tracing data, invasive track tracing data from Henry Kennedy's lab, based on the pattern of proportion of connections that come from the superficial layers to the deep layers. You can estimate a kind of cortical hierarchy. And we can see that they pull out basically the same gradient that we see in the receptor organization, saying that there's this strong relationship between the receptor organization, the dendrites, the interareal connectivity, and the myelin across the cortex. So more generally, um, um, kind of pulling out a bit, I think that as we can understand this as potentially a kind of a gradient of flexibility. In the sensory networks, we're seeing large density of neurons, but lots of myelin, also lots of what we call parvalbumin cells, which I haven't mentioned yet, which are all thought to be kind of markers of some sort of stability in a network. Whereas as we go up to the other end of this gradient, we see more neuromodulatory receptors, more inputs, bigger dendrites, less myelin. So more of these kind of markers of flexibility. So we might be thinking that this sensory to cognitive hierarchy might also be reflective of a kind of a hierarchy of flexibility along the cortex that is enabled by these different factors. All right, I'm just going to take off my jumper for 1 sec. Sorry, the talk's really heating up. So. Okay, so I'm really interested in how anatomy enables and constrains cognition, right? So which aspects of our anatomy enable us to think the way we do and perhaps differently from different animals and so on. Okay, and to do this, we wanted to develop a kind of computational model that would allow us to zoom in on what's happening at the level of neural circuits, because we think a lot of this neuromodulation and the computation is happening at the level of neural circuits. But we also wanted to be able to zoom out and to try and understand how this affects these large-scale cognitive networks during cognitive tasks. Okay, so and of course there's other people doing related work. Um, we developed this multiscale brain network modeling of cognition. Okay, I'll point out, for example, Jorge Mejias has also done relevant work in this area, and I think there's some recent work from Gustavo Deco's lab as well. Okay, but I'm going to take you through the basic building blocks of how we build one of these models, right? And this is actually not from my work as such. This is from the first um, uh, kind of foundational paper that really inspired me to move in this direction, which is from Rishi Chowdhury and Xiaojing Wang. Okay, and so this is not one of these whole brain models that really recently came out from John Fang Fang's lab with 86 billion neurons. No, this is much, much simpler. Okay, so we can go through it in just three short steps. All right. So the idea is that in each brain area and each cortical area, usually, you just have one simple local circuit model. Okay, and in this case, it can be as simple as two dynamical equations, two ODEs, one representing the excitatory population, one representing a population of inhibitory neurons, and they interact. Okay. And in this paper by Rishi, what you can see, Rishi Chowdhury, what you can see is we're trying to understand with this equation how the firing rates of the excitatory populations, shown here with E, in this particular area X, how they change over time in response to different inputs. Okay. All right. So if we just, what we're going to see here is the different synaptic inputs. But first, let's just look at this. This just says that, okay, if the activity is positive and it's always non-negative, then if there's no synaptic input, the activity is going to decay back down to zero. Cool. And this little tau, that's just the time constant. That's going to say how quickly your activity is going to decay back down to zero. Okay. But obviously the interesting stuff happens when there is synaptic inputs, right? So it's super simple. We just say that, all right, we have this the activity in this area, these excitatory neurons, it goes up when the firing rate in these same excitatory neurons is high, and it does so proportional to the strength of this kind of recurrent excitatory connectivity from the excitatory population to itself. Okay. And then it goes down when the inhibitory neurons are firing a lot. And it does that in proportion to this input from the inhibitory to the excitatory population. Okay. This tiny little plus that just says that firing rates don't go below zero. So we just make sure that that if anything is going to go below zero, we just set it at zero. And this is just a constant, okay, to keep it in a kind of a realistic range. Okay. Are we good? Yeah. Okay. I'm going to take silence as approval. But okay, cool. Um, all right. So this is just a local circuit.
Not just silence, also nodding, so you can
Oh,
see.
nodding. Yeah. Okay. Cool, 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 cool. All right. All right. So, all right, that's the local circuit model. Okay. But we want to connect these blocks together, right? And this is where groups like Gustavo Deco, his lab, they really started doing this. And Michael Breakspear, beautiful stuff. The idea is that maybe we can try to understand the dynamics, not just of one area, but across kind of, dozens of areas across the whole cortex. So to do that, you want to connect these different building blocks according to the strength of the connectivity between areas. And if you're simulating human data, you can do that with diffusion MRI tractography. But as we're working with animal data here, we can use this beautiful invasive retrograde tractography data. And to do that, we teamed up with Henry Kennedy's wonderful lab in Lyon. Okay, so how do we do that? It's super simple, right? We just say if we want to understand how inputs from other areas affect our firing rate in this area, we just say, okay, if those excitatory neurons in any other area Y are firing a lot, that's going to affect our firing rate according to the strength of connectivity from area Y to area X. And we just sum that up over all of the areas, and this G just tells you how strong the long range inputs from other areas are overall in the model. Okay. Um, Anything I'm missing? Oh yeah. So we only care about excitatory neurons here because generally in the cortex, of course, biology is weird. There's always exceptions, but in general, it's only the excitatory neurons that project long range between areas. All right. Cool. Okay, but this is the step that was, I think, original to this paper, and it got me kind of really excited about thinking about how we might get towards different functions emerging from different parts of the cortex. Okay, and what Rishi Chowdhury and Xiaojing Wang said was that, well, you know, all different parts of the cortex seem to differ, not just in respect to their long-range connectivity, but also local circuit properties that are intrinsic to each area. Okay. And here we see this data from Guy Elston again with the dendritic spines, and he's noticed that the density of spines. So the spines are the location on the dendrites where the excitatory synaptic connections come on to other excitatory neurons. Okay, so you can kind of think of this as synaptic excitatory connections, and this is on pyramidal cells, okay, and an individual pyramidal cell across these different areas. And what you see is that this increases um, about tenfold as you go from V1 up to higher parts of the temporal cortex and prefrontal cortex. Okay, so Rishi and Shaojing was like, well, maybe this is important. Okay, and so how do you put this into your equation? It can be pretty super simple. You just take your spine data here, Sx, okay, and you just basically pop it in front of your equation so that you scale the total inputs to an area in proportional to the spine count, in proportional to the kind of number of synapses per neuron in the area. Right, cool. I probably don't have time to kind of go into it, but there's some beautiful predictions from this paper about a hierarchy of timescales emerging in the cortex that has then since held up really nicely and been kind of shown to be true across species. Okay, right, cool. So we took this basic approach and with slightly different local equations. So now it's nonlinear and you can get kind of some slightly more interesting dynamics. Okay, and we wanted to um, simulate this task. What task? Well, I want you to put your little thumb up if you can whenever you see the circle on the screen. Okay, okay, cool. Are there any virtual thumbs up or no? Okay. So maybe it's hard to do on the new Zoom. I'm not sure. Okay, Petra saw the circle. All right, cool. Ah, thank you, Rachel. Nice one. All right. Okay, okay, lots of thumbs up. It's coming. It's really there. It's not a trick. Okay, nice. All right. So can you see the circle? Okay. Some of you can see it. That's great. In reality, um, researchers actually do this type of experiment all the time when they're trying to find something no less grandiose than the neural correlates of consciousness, or I should really say the neural correlates of conscious access, right? So they show difficult to see stimuli like this very quickly, okay? And you can taper the strength and the timing of the stimulus so that they can show you this 100 times and you'll notice it about 50 times and the other 50 you'll think it wasn't there. Okay. And the nice thing about this is that then you can compare the brain activity for exactly the same stimulus on the trials when it was perceived versus the trials when it wasn't perceived. Okay. And by doing the subtraction of the activity on the non-perceived trials from the perceived trials, researchers think this is a putative neural correlate of conscious perception. Okay. Then they can also be really sneaky sometimes, and they can say, okay, we're actually not going to show any stimulus, and we're still going to ask you if you've perceived it or not. Okay. And and uh, and this can kind of allow us to differentiate these trial types. Right. Okay. So what's interesting about this type of data then is whether the cortex principally encodes the objective reality of whether a stimulus is there, or whether it is encoding more strongly the subjective perception, or like, do you see high firing rates when the activity was perceived and low otherwise, or is it high firing rates when the stimulus was really there and low otherwise? So can I have a little vote with virtual thumbs up if people think that the cortex is primarily encoding the objective reality of whether the stimulus is on or not? Okay. Okay, one vote. Okay. All right. What about who thinks that the cortex is primarily encoding the subjective experience? Like, okay. All right. Well, I think the 10 thumbs up from Petra kind of really wins that vote there. Okay, cool. All right, so this is the experiment from Van Vugt and colleagues, Peter Rolfson's lab a few years ago. They did exactly this type of experiment in monkeys and they put their electrode in V1 primary visual cortex. And um, and they had these four different trial types, and what you can see is really the principal driver of firing rates is disappointingly not the subjective perception. It's high on these green and red trials, so it's high whenever the stimulus is there, and it's low otherwise. Okay, so it seems like V1 is more closely corresponding to an objective encoding of the stimuli. Then they moved their electrode up to prefrontal cortex. And you see a very different story emerge after about 200 or 300 milliseconds. Okay, you see this high activity that is actually sustained for a few hundred milliseconds emerge in dorsolateral prefrontal cortex. Whenever the stimulus is perceived, it's actually kind of not so strongly affected by whether the stimulus was really there or not. And around the same time frame, when the stimulus is not perceived, you see this low activity. Okay. So along the cortical hierarchy, there seems to be some sort of a shift from primarily encoding the objective stimulus to the subjective experience. And Elise, who's a wonderful PhD student working with me and Daniel Margulies, he decided he wanted to try to simulate this in a computational model. Okay. And if you zoom in on area V1 and DLPFC in his model, you see this these activity patterns for the four different trial types that look, really quite similar to what Van Vogt and colleagues saw experimentally. So, kind of a high activity whenever there's a stimulus in V1, and this slow kind of binary activity after a couple of hundred milliseconds in DLPFC on these hit trials, hit and false alarm trials. Okay. But the cool thing about our model being multi-scale is then we can zoom out from these areas and we can look at activity across the entire cortex. Okay. And what we can see here on the top is activity during a hit trial, and on the bottom is activity during a mistrial. I'm not sure if there's a bit of a problem rendering there. But basically, the principal difference here is that this kind of strong, sustained activity that experimentally they saw in DLPFC, we actually see through a distributed network of regions that we think roughly corresponds to what human cognitive neuroscientists call the frontoparietal network and the dorsal attention network. Okay. All right. Ulysse then simplified this big multi-area model just down to two dynamical variables to try and understand the dynamics a little bit. And and we have this phase portrait with these node lines here, and the node lines just tell us that if you're on this particular point on this particular line, your inhibitory activity variable is not going to change in time. And if you're on this line, your excitatory activity is not going to change. So when these two lines cross, we have the stable states of the system. Okay. Sorry, we have the steady states of the system. Okay. And we see here we have two stable steady states and one unstable steady state. Okay. An unstable steady state is one where if there's no noise, your system activity can stay exactly there. But as soon as there's a slight deviation from that, it's going to be repelled away from there. Okay. And the stable steady states are also called attractor states. That means that anything that is within the kind of orbit of that activity state is going to be pulled towards that. Right. What we see at the start of the trial here is that activity starts low, close to this low activity attractor state. And then when the stimulus comes on, we see this null climb pushed up, right? So there's this kind of dynamic bifurcation. Bifurcation is when there's a change in the number or stability of steady states of the system. And we see that this is pushed up, so activity now rushes up towards the one remaining steady state, the one remaining attractor. Okay, but the stimulus is only very brief. So as soon as you remove the stimulus, this null climb comes back down. And what determines whether the activity will go up to this high activity that you need for this high attractor hit or low attractor, go back down to the low attractor, is whether this null climb comes down and the activity is to the left or the right of this unstable steady state, which acts as a kind of a threshold. Okay. So it's this. So we understand this stimulus perception across the cortex according to this kind of simplified dynamical system. Okay, and this dynamic bifurcation mechanism. Right, cool. And here we can see the same thing playing out across the whole cortex and a miss and a hit trial. Right. Right. So we try to get many of the parameters for this types of model from data, right? We have the connectivity, we have the spines, we have some we set the synaptic weights according to physiology data, but there's certain parameters that we just don't know. We couldn't find experiments to be able to set them. They haven't been done yet, and critical parameter here is whether the feed forward connections, so those connections that go up from the sensory parts to the kind of higher cognitive parts of the brain, whether they principally work via fast or slow excitatory glutamate receptors. The fast ones are called AMPA, the slow ones are called NMDA. And we also wanted to know in the feedback direction. So also you can think about that as like a top-down influence from higher areas like prefrontal cortex to the sensory areas. Do they use the fast or the slow, the AMPA or the NMDA glutamate receptor? Okay. So in our model, we replicated not just this one result I mentioned from Van Vogt, but actually the same model was able to also capture results from Claire Serjeant's lab doing experiments with human, as well as some results from Stan Dehaene. So but for all of those three experiments that we were able to replicate, we needed to have the model in this particular part of parameter space. And this particular part of parameter space had the feed-forward connections being mostly AMPA, these fast glutamate receptors, and the feedback connections being both mostly NMDA or slow glutamate receptors. This is a bit surprising, okay? Because it makes this prediction that the NMDA AMPA ratio, here we call it the NMDA fraction, actually goes down along the cortical hierarchy. Now, why is that surprising? Well, people have previously emphasized how important NMDA should be for prefrontal functions like working memory. And they've also thought that, well, in sensory cortex, you may want really fast processing, so you may want to use AMPA. And we're actually seeing this kind of seemingly opposite trend emerge in our models here. So we were like, okay, that's a bit surprising. Let's team up with Nicola Palomero Gallagher again, because we can check that directly in the data. And when you look at this in vitro receptor radiography data from across the cortex, you see this very similar decrease in the NMDA fraction or the NMDA AMPA ratio across the cortex. Okay, so this is a kind of anatomical discovery that that we never really would have even thought to look for if we didn't have a model that was capable of linking from receptors through cortex-wide dynamics and cognition. And we actually think that this decreasing NMDA AMPA ratio along this cortical hierarchy is actually required to enable rapid cortical ignition during conscious access. Cool. So this is kind of related to the global neuronal workspace theory from Stan Dehaene and colleagues, as well as previous versions from Bernard Bars and so on. And they put forward this idea that you have this strongly connected set of neurons across the cortex, and that they're going to call the global workspace, and different systems, perceptual systems, memory systems, etc., are all competing for access to that kind of strongly connected network. Okay, and I think that's broadly consistent with what we think the result of this work. But there were some at least versions of this global neuronal workspace theory that emphasize that sensory activity is actually maintained for that information to come into kind of conscious awareness, conscious perception. And what we see is a slightly different thing, at least in our modeling, and I think it's consistent with the data, that you see this kind of wave-like propagation of sensory information from the early sensory cortex into the attention and frontal parietal networks. And it's when the sensory information is sustained within these attention and frontal parietal networks that we start to, we think information will reach conscious awareness. Okay. Um, okay, cool. How are we doing, Petra? Crystal, I see there's something in the chat, but I haven't seen. Is that a question that I should deal with now?
I know nothing for you to deal with in the chat, and
Okay.
really, really interesting so far. So please carry on. You've got, yeah, there's about 20 minutes left, and you can present more or leave more time for Q&A, whatever
Yeah.
works.
Cool, cool, cool. Okay. That was very nice, Petra. You kind of had to say that. All right, cool. So I started talking about neuromodulators early, and I really wanted to get back to that. Okay. And I'm going to focus here on dopamine for a few minutes because of all of the 14 different types of receptors that we analyzed, dopamine was the one that was most strongly correlated with this sensory to cognitive hierarchy. Okay. And we're interested in analyzing how dopamine across the cortex may influence working memory. So there's old studies from Paco and Rakij and others showing that if you remove dopamine from the cortex, it can have hugely debilitating effects on working memory. But most of these studies have focused on dorsolateral prefrontal cortex, so quite a restricted part of cortex. And now we see that this receptor pattern is actually quite distributed in this kind of gradient. So we wanted to understand how this dopamine affected this wider network and that we now know contributes to working memory. So again, we developed one of these multiscale models of the macaque cortex. Here we have a slightly more intricate local circuit. So we have, in each brain area, we have two competing excitatory populations shown here in red and blue. And we have three different types of inhibitory neurons. We have the PV interneurons that inhibit the cell body area of the pyramidal cell. We have the SST interneurons that inhibit the dendrite, and they themselves are inhibited by the VIP interneurons. Okay. And then we applied the dopamine effects in the model based on multiple neurophysiology studies, mostly slice physiology. And this had three effects. One was that as dopamine increased, the strength of excitation via the slow excitatory NMDA receptor increased. The second effect was that the target of inhibition shifted from being primarily towards the soma to being more towards the dendrite. And then for very high levels of dopamine, we started to get an increase in these adaptation. As with the previous models, we also implemented this increase in the spine count and the connectivity data. All right. So we tested this model on a range of simple working memory tasks. And for these tasks, we present it with a stimulus, right here shown in red as the target stimulus. And you can think about that as activating the red excitatory population that we showed on the last slide. And we present that just to the excitatory cells in V1 at the back of the brain here. Okay. And then we want the network to hold on to the memory of that stimulus through until the end of the trial. So there's a delay period when there's no stimulus. And then during the delay, we're going to put a distractor stimulus that we're showing in blue here. Okay. And you're going to see that we're, by having these two populations in each area, we're able to dissociate the activity of an area from what it's representing. Okay. And we're going to show activity representing the target stimulus in red and representing the distractor in blue. So at the start of the trial, activity comes in through the sensory cortex and it propagates through the visual streams up into the frontal parietal network. During the delay, you see that activity is dying off in these sensory areas, but it's maintained in this frontal parietal working memory network. Okay. When the distractor comes in, it propagates forward and it tries to dislodge the memory of the original stimulus from the frontal parietal network, but in this case it fails. We see that we still have red activity at the end of the trial representing a successful distractor-resistant working memory. Okay. So now we have these distributed working memory patterns. We need to know whether that's a reasonable pattern or not. and Julio Martinez Trujilla's lab had just done this big mega analysis of over 90 working memory studies of in vivo electrophysiology in the macaque monkey. And this is the pattern of activity that they saw in their studies with this working memory delay activity, usually seen in these green areas, but not the gold areas. And this was the activity pattern we saw in our model. Okay, so it seems to match really nicely, and this is really just from implementing these different features of the anatomy, the spines, the dopamine, and the connectivity. Another aspect of the model is that we were able to shift the network from being distractible to distractor resistant just by shifting the level of dopamine in the cortex. And by having this kind of a multiscale model, we can then ask, well, how can that happen? Right? So we were able to zoom in on what was happening in individual cortical areas and these cell types. And we found that if we simulate the optogenetic inhibition of these dendrite targeting inhibitory neurons at the moment of the distractor, that can switch the network back from being distractor resistant to distractible. So to sum this up, our conclusion was that the gating of information entering this frontoparietal working memory network, this occurs via these dendrite targeting SST inhibitory neurons, and that's facilitated by the release of dopamine, such that it's occurring in areas with high levels of dopamine D1. Okay, so it's pretty specific prediction. And there's this recent preprint that just came out from this group in Sweden that did slice and in vivo experiments, actually in vivo experiments principally, to test this idea in a different task. And they concluded that prefrontal gating is mediated by the recruitment of dendrite targeting somatostatin interneurons and facilitated by dopamine. So it's nice to see some sort of a kind of independent experimental support for this idea, but the dopamine literature is complicated and messy, so I think it's still to be determined. All right. So this is the main part. We're going to try and fit in a little bit of a next part if we have time. But so the idea from this first part is that we have this primary gradient of receptor density that goes from sensory to cognitive areas. It enables this rapid, reliable propagation of information across from the sensory cortex up the hierarchy, and also the flexible selection of information entering the higher cognitive networks through this distribution of D1 receptors. All right. But you notice I showed there was two major axes at the start. Um, and so let's dig in quickly. So there's time for questions into the second gradient. All right. So the second gradient, if we we try to understand that by looking at areas that have a similar value on this first principal component, this first gradient, but very different values on the second axis. And what we see here on these kind of polar plots, where the closer the line is to the edge, it indicates a higher density of receptors. We see that the overall size of the shape is similar, indicating that they have a similar number of receptors in total, but the pattern is very different from one axis to another. And this is principally driven by the serotonin receptors, and primarily the serotonin 5-HT1A receptor, which is very low down this end and very high up this end. Okay. Um, but you'll notice also that a lot of the areas in this kind of top right corner are red, and that's because they're colored because they belong to the default mode network, which we think is mostly active for perceptually decoupled or internally driven cognition, things like imagination, autobiographical memory, recall, and so on. And a lot of the regions down here in green are belong to the default, the dorsal attention network, which is active for kind of these opposite situations where your attention has to be on the external world, external stimuli. So it's interesting that just by looking at the anatomy, the receptor expression, we see this kind of opposition between these internally and externally focused cognitive networks. Okay. So just by looking at the anatomy, we thought that, well, maybe then serotonin, which really drives this axis, may bias the competition between internally and externally focused cognition. Okay. So far we've talked about how sensory information can perhaps reach our conscious awareness. But of course, a lot of the time, about half of the time, our mind is on something not sensory driven at all. It's on our internal thoughts. So how does that reach our mind? And so we don't know, but one possibility is that internally driven information has to propagate from regions like the hippocampus and parts of the default mode network and compete for access to the same frontal parietal network in order to capture our minds. And there's been some beautiful work recently in individual level of fMRI that has shown that actually this frontal parietal network can be separated into two subnetworks. One that is more strongly connected, functionally connected to the default mode network, and the other that is more strongly connected to the dorsal attention network. Okay. So in theory, if we look at just activity in this frontal parietal network, and we consider the pattern of activity in these frontal parietal network areas as a vector in activity space, there's going to be one direction that is aligned with this internal attention default mode direction, and another that is aligned with this dorsal attention, external attention direction. And by looking at the pattern of activity, how it changes in time, we should be able to see how this activity aligns with these two directions and therefore predict whether the mind is focused on external stimuli or internal thoughts. Okay. Um, and we've developed these ideas in a new computational model. And the idea is basically quite simple, right? Because we see in practice, decades of studies have shown that there's often an anti-correlation between this default mode and dorsal attention network. And to try to explain this from an attractor type perspective, we've come up with a very simple proposal. Is that the default mode network and the dorsal attention network um, are both um, connected a little bit too weakly so that they're not individually capable of sustaining a high activity attractor state. So in order to do that, they need to recruit parts of the frontal parietal network so that we actually see competition at the level of the frontal parietal network between those sub networks that connect to the default mode or the dorsal attention network. And whichever subnetwork wins that competition essentially will dictate the contents of whether your mind is on internally generated or externally generated thoughts. I'm just going to skip over the details of this for time here. There's lots of cool stuff in this if you want to dig in. We see chaotic attractors and also decodable activity. So yeah, check it out if you're interested. Okay. So the idea is basically these default mode and dorsal attention networks compete to form attractor states with the frontal parietal network. And more speculative bit is that actual competition may dictate whether our awareness is oriented towards the internal thoughts or the external sensory world. Okay, right. Cool. So how can we test this a bit more directly, or how can we understand whether serotonin is really driving that competition? All right. So it's pretty difficult to do these studies in primates. So then we looked and we found that the pattern of this serotonin 1A receptor is actually pretty similar between humans, macaques, and rats. And it peaks in all species in these purple areas here, which are the anterior cingulate, medial prefrontal cortex, which we think is part of the default mode network. Okay. So we said then, okay, so why don't we try to understand what's happening locally in this area with super high serotonin 1A receptor density receptors when we give a drug that acts on the serotonergic receptors. And so we did this experiment. Well, I didn't do it. My collaborators did, Ross Purple and Matt Jones, and they did neuropixels recordings from across these medial prefrontal and anterior cingulate areas during different doses of psilocybin or a saline drug. Psilocybin or magic mushrooms acts on the serotonin 5-HTA, 2A, and 1A receptors. Okay. What we found in this area, um, somewhat surprisingly from compared to previous results, is that psilocybin had primarily an inhibitory effect. We see it in the cingulate and also the other medial prefrontal regions. And with Rahul, a former postdoc in my lab, who's now got a faculty position in India, he found that when we look at the mean squared displacement of activity over time, you can think of that as the kind of the speed of the neural dynamics over time. This kind of systematically decreased with psilocybin. Okay. So in this part of the cortex with a lot of the one serotonin 1A receptors, we see that psilocybin inhibits activity and slows neurodynamics. Okay. And I think that actually makes sense because a lot of the other cortex, parts of cortex have huge amounts of the serotonin 2A receptor, which has this excitatory effect, whereas 1A has this local inhibitory effect. Um, so, although this might be of interest to the psilocybin literature, for our story, this might give us a hint at how the serotonergic system might be biasing competition between these larger cognitive networks. Okay, so this to kind of put together all of these different ideas. That this idea that serotonin may dampen activity in the default mode network. We've seen that in the medial prefrontal cortex, which is this keynote of the default mode network, right? And by doing that, it might bias the competition between the default mode and dorsal attention networks. It might do that in favor of these external attention and sensory networks. Okay. All right. So to conclude, we see this primary gradient of receptor density. We looked at NMDA, AMPA, dopamine. We saw how that can help information propagate from the sensory areas and help the selection of information entering higher cognitive networks. We've also seen a secondary gradient really driven by the serotonin receptors, particularly 1A, and we are, developing these ideas that this may help to switch our between higher cognitive networks and do that by biasing competition between these distributed multi-area attractors. All right. Okay, so bring it back and let to bed. The idea is that we can have these different attractor states or these different kind of easy to get into patterns of neural activity in our brain, but we can actually change which are the easy patterns of activity to get into at different times by giving in different uh, um, neuromodulatory inputs to the cortex. Okay, so we can think about that this subcortical centers can control the neural dynamical landscape, and they can do that by activating these gradients of receptors. Thanks so much. I have lots of collaborators. It's a big team effort. Really appreciate the invitation and your time. Thank you.
Amazing. Thank you so much. That was a fantastic talk.